टॉपिक है टॉपिक इज रिकंस्ट्रक्टिंग बायोलॉजिकल डाइवर्सिटी. This means that we are studying biological diversity in its holistic view, and we try to find out relationships among different kinds of organisms, how they are related, how they are different, and where they differ, why they differ, and where from the present day plants and animals and all other kinds of organisms have come from. When we talk of evolution, all plants, animals and other organisms which are alive today, they have descended from earlier organisms by genetic modifications, and these modifications have accumulated in successive generations, and finally the ancestral populations or generations have divided into different kinds of organisms. That is, these earlier organisms, they descended even more from ancient forms, and these from yet form older forms of life. That is, these earlier organisms descended from even more ancient forms, and these from yet have in turn evolved from older forms of life, and this chain of descent continues back in time to the beginning of life. That is where from actually life has initiated. Present day organisms have their story in this descent relationship, that is ancestor progeny relationship. These evolutionary forces that often get major consideration because it leads to adaptation. It is the natural selection. What is natural selection? Nature selects those which are fittest, that is fittest can survive and they can win the race. For example, you were 33 students in class 12th, and among those 33 students, only 2 or 3 have qualified NEET and they are pursuing MBBS. Among those 33, 20 are pursuing graduation, and rest have left their studies. This is selection, and this way nature too selects those organisms which are successful and adapt with the changing environment. They continue their race. These offspring which inherit the successful adaptations and thus also leave more offspring. Here it is biological potential, reproductive potential. That is, those organisms which have higher reproductive potential and can adapt to the changing environmental conditions, they leave more offspring, and those organisms continue their race. Eventually, after several generations, adaptation becomes a feature of all members of species. Here we see it is the adaptation that helps in evolution. जो natural world है, the natural world before us, it can be explained. Similarities among organisms are due to common descent, and differences are due to adaptation to environment or less commonly chance events. यानी जो similarities है organisms में, for example, we find similar proteins throughout the living world. We find similar DNA, we find similar RNA, we find several other similarities, and this tells us that all organisms have evolved from the common ancestor, that is, they have common descent. And the differences which we see among organisms, जो आप differences देखते हैं, it is due to adaptation. यानी various organisms have adapted variously, and those organisms which could not adapt to the changing environment, they succumbed, they were defeated in the race of life, and they were eliminated. This is the natural world. That is, similarities among organisms जो है, they are due to common descent, and differences are due to adaptation to environment. Differences जो है, they have come as a result of adaptation to the environment. They have come as a result to the to adapt to the varying environments, which we also called chance events. As the environment never ceases changing, yani environment continuously changes, animals and plants accumulate modifications and are continuously molded into entirely different forms and are continuously molded into entirely different forms. दिस इज इम्पॉर्टेंट है, इसको मैं फिर से रिपीट करूँगा. द नेचुरल वर्ल्ड, जिस नेचुरल वर्ल्ड में हम रहते हैं, particularly the living world, we can put it in these lines. That is, similarities among organisms जो है are due to common descent, are due to common descent. That is, whatever similarities we see, it is because we have come from common ancestor, and the differences which we witness, और जो differences हम देखते हैं, they are due to our adaptations to the environment differently. यानी some organisms could adapt this way and some organisms could adapt that way. For example, Darwin, when visited the Galapagos Islands, he observed the finches there, and they had differences in their beak sizes, and he explained those differences in beak sizes due to their different adaptations to food habits, and those beak sizes were, or beak shapes were, due to various adaptations to varying environmental conditions. Now, systematics. What is systematics actually? It is the determining the pattern of evolution. Perhaps Darwin's best argument that life evolved was observed hierarchy of the life. All organisms are united in a pattern of increasing similarity. This is important. यानी जितनी भी organisms है, we have a pattern of increasing similarity. The pattern of this relatedness, that is, we are related, it shows phylogeny. Phylogeny means evolutionary history. Evolutionary history. That is why we are related, because we have an evolutionary history. We have come from common ancestor. We have same descent. Our ancestors were same, and that is why there is relatedness among different kinds of organisms. For example, when we see the forelimb of human beings and the forelimb of a bird, it has same structural plan, it has same skeletal plan, except birds have feathers and human beings do not have feathers, but the anatomical plan is same. This relatedness tells us about the phylogeny. Phylogeny का मतलब है evolutionary history, and systematics is the field of biology that studies and seeks to determine the phylogenies. यानी systematics का real essence जो है या actual purpose जो है, it is that field of biology which studies and seeks to determine these phylogenies. Evidence used to determine phylogeny, यानी कौन से evidences है, कौन से सबूत है, which proofs are there that are used to determine phylogeny, that is evolutionary history. जो data systematists है, those which accumulate data, we deal with the data and use the data to reconstruct phylogenies. We call these scientists as data systematists, and they data systematists use to reconstruct phylogenies. That is, phylogenies are constructed, evolutionary histories are constructed. That is where we have departed from apes, or where from bryophytes pteridophytes have evolved, and where that branching has occurred, where from advanced pteridophytes primitive gymnosperms have evolved, and this branching pattern or this evolutionary history, this is constructed by data systematists by the use of characters or data that organisms have. What are those characters? These characters, for example, the character of human beings is nose shape. On the basis of nose shape, we can group human beings into those which have longer noses and those which have shorter noses. On the basis of skin color, we can say that these are gore, these are kale, like these are black colored people, these are fair colored people. This is the use of characters in grouping of organisms. Likewise, the data systematists reconstruct phylogenies, and they group organisms on the basis of these characters. And what are these characters? These characters come from morphology. When we talk of morphology, morphology is simply the study of structure and form. यानी structure क्या है, और फॉर्म कैसी है, शेप कैसी है, दिस बिकम्स द मॉर्फोलॉजी. फॉर एग्जांपल यू स्टडी इन plant morphology, the shape of root, the shape of stem, the type of inflorescence, the type of flower, the number of petals. These are the patterns which you study in morphology. The phyllotaxy, the number of leaves, the number of the shapes of leaves, the number of leaves. These form the aspects under morphology. There and we have developmental biology. Developmental biology is that biology which is used, which concerns with the development of an organism, where from the organism has evolved. It has evolved from a single cell, that is zygote. How zygote has come into existence? It has come into existence by the fusion of male and female gametes. Where from those male and female gametes have come? They have come from the parent organism due to megasporogenesis and microsporogenesis. As a result of microsporogenesis, microspore has formed. Microspore has finally evolved into are formed into pollen grain, and pollen grain contains male gamete, and megaspore has formed into embryo sac or female gametophyte, and that female gametophyte contains the female gamete or the egg. By the fusion of male and female gamete, zygote is formed, and by the various morphogenesis and other changes in this zygote, organs have formed and an organism has evolved. This all we study under developmental biology. The third characters we take from molecular biology. Molecular biology includes genetics, it includes biochemistry, etc. etc. When we see the developmental patterns same in organisms, and when the developmental patterns are similar in organisms, groups of organisms, we say that organisms are related, or when they are different, we say that organisms are different. Likewise, we use molecular biology, we use DNA, and we use RFLP, restriction fragmental length polymorphism, AFLP, etc. etc. of this genomics, which help us in identifying the closeness or relatedness of organisms. In phytochemistry, we use the biomolecules, we use the chemicals and the relatedness in chemicals to decide whether organisms are related or not. In morphology, what we do, morphology में क्या किया जाता है? Systematists study external characteristics, examine bones and teeth, dissect organ systems, make histological light microscopic slides, and peer at the cells, look at the cells and tissues with electron microscope, all to gather the comparative evidence for evolution. That is, to find out similarities and differences. And under microscope, if we see the relatedness among organisms in mitochondria, in chromosomes, in genes, etc., we can conclude that organisms are related. Along the way, much is added to our knowledge about the basic biology of different organisms. In developmental pathways, developmental history is called ontogeny, and evolutionary history is called phylogeny. In developmental pathways, what we do, the basic embryonic development जो है, embryonic development, how embryos are developed, it is constrained and really changes. It does not change. It is similar. It follows a pattern that is not subject to change and really changes. And because of any mutation that affects this developmental pattern, that is the formation of embryo or the ontogeny, in the early development is almost always lethal and can cause the death of an organism during its development and selected against, and that is those organisms which succumb during the embryonic development, that due to some changes which are lethal to the organism during its development, those organisms are excluded. They do not continue their race. Therefore, developmental evidence can be particularly useful in uncovering evolutionary relationships between diverse adult forms, which we study under as embryological evidences of evolution, morphological evidences of evolution, paleontological evidences of evolution, ontological development or developmental evidences of evolution, etc. etc. Molecular and biochemical data, because the characteristics of an organism are determined by its genetic content, that is its gene pool, that is the number of genes. Changes in organism over the course of evolution should be reflected in their genetic information, because we know the central dogma of molecular biology, wherein DNA forms RNA and RNA forms protein. Proteins give the phenotype to an organism. Thus the phenotype of an organism, or the shape of an organism, or the morphology of an organism is hidden in these genes. This is according to central dogma, and the differences in phenotype of organism is thus contained in the genotype of an organism, and the relatedness in genotype would be a useful tool in tracing the evolution, in deciphering the evolution, in determining the phylogeny, that is the evolutionary history. Caution is needed though. Similarities may not always be as a result of common ancestry. Similarities may be otherwise also. Molecules and genes could have similar construction as a result of convergent evolution as well. For example, we see a bird and an insect both fly in the air, an insect as well as a bird both fly in the air, and we say that they are similar. No, they are very diverse. The wing of a bird and the wing of an insect are similar, but they are analogous. They have different anatomical plans. Wing of insect is similarly a subcutaneous appendage, an epidermal appendage, while the wing of bird has a anatomical plan. It has bones, it has shorter bones, it has longer bones, it has feathers. So apparently they look similar, but at anatomical level they are not similar. At anatomical level they are not similar. This means that similarities are not always due to a result of common ancestry. It could be sometimes misleading as well. Analyzing the data to reconstruct evolution, यानी हम कैसे डाटा इस्तेमाल कर सकते हैं एवोल्यूशन को कंस्ट्रक्ट करने में. Do you listen to me? Am I audible?
Yes, sir.
Am I audible?
Yes, sir.
सबक आ रहा है समझ सारा? समझ आ रहा है सारा समझ?
Yes.
अरबी ना अटेंडेंस ले रखना, अटेंडेंस. अच्छा, now we have part second. Mute रखो, mute. Part second. Analyzing the data. सारे रखो mute. Analyzing the data to reconstruct evolution. यानी how we can analyze the data and how we can reconstruct the evolution. When we see how Darwin came to his famous theory of evolution, that is which is contained in his book Origin of Species, नेचुरल सिलेक्शन पे वो कैसे पहुंचा, सर्वाइवल ऑफ फिटेस्ट उसने कैसे दिया, हाउ ही केम टू conclusion that those fishes had common ancestor, and we can say that he simply reconstructed his data, he simply analyzed his data and constructed the theory of evolution, constructed the hypothesis of evolution. There are three major ways in which classifications are constructed. We can construct classifications by three ways. Number one is phonetic or numerical taxonomy, the second is synthetic or traditional taxonomy, and the third is cladistic or phylogenetic taxonomy. In cladistics we construct phylogenetic trees, and in traditional taxonomy we do everything. We include morphology, we include anatomy, we do traditional botany, etc. And in phonetics we do numerical taxonomy. Phonetics or numerical taxonomy, many people of you have problem with this numerical taxonomy or phonetics. Phonetics or numerical taxonomy, this method that uses overall similarity among organisms, we call it phonetics or numerical taxonomy. How we construct this overall similarity? We take larger number of characters, and those larger number of characters, then we do comparisons among organisms, and those organisms which have larger number of characters in common are put together and are grouped under one cluster that we call clustral analysis. In measuring similarity, phoneticists जो है जिस तरह डाटा सिस्टमैटिस्ट्स थे जो डाटा इस्तेमाल करते हैं फॉर कंस्ट्रक्टिंग एवोल्यूशन, इसी तरह फिनेटिस्ट्स जो है और न्यूमेरिकल टेक्सोनमिस्ट जो है, दे यूज ए वैरायटी ऑफ टेक्निक्स एंड मेथड्स एंड all though they use variety of techniques and methods, but all use following steps. What are those steps in numerical taxonomy or phonetics? Number one is a data matrix is constructed. What does this data matrix mean? It is constructed and it lists character status in coded numerical form for all characters among all of the organisms or taxa under study. I will explain this. यहाँ पे आप ये याद रखें कि एक डाटा मैट्रिक्स बनाया जाता है जिसमें लिस्टिंग द कैरेक्टर स्टेटस, यानी एक कैरेक्टर के बहुत सारे स्टेटस हो सकते हैं. फॉर एग्जांपल स्किन कलर के आपके बहुत सारे स्किन कलर में शेड्स होते हैं. दोज आर स्टेटस ऑफ स्किन कलर. फॉर एग्जांपल आपने वो फेयर एंड लवली का ऐड पढ़ा होगा जिसमें गोडनिक दो ये फेयर एंड लवली मथन तम दो अख शेड आसन, दूसरे दिन तीसरे दिन चौथे दिन पाँचवें दिन 10वें दिन और लास्ट दिन कितनी फेयरनेस आ जाती है बाय रबिंग दैट फेयर एंड और यूजिंग दैट फेयर एंड लवली. जो वो डिफरेंट शेड्स ऑफ स्किन हासिल होते हैं आफ्टर अप्लाइंग दैट फेयर एंड लवली फॉर मैनी डेज, दोज आर सिंपली द कैरेक्टर स्टेट्स. फॉर एग्जांपल वी हैव सिस्टम हाइट, देयर कुड बी सेवरल हाइट्स ऑफ सिस्टम. एक ज़रूरी ऐलान, अरबीना.
जी सर.
आफ्टर दिस क्लास यू विल ज्वाइन अगेन यूजिंग द सेम लिंक.
ओके सर.
क्योंकि ये कंप्लीट करेंगे. टैबसिटन माकली जो आपने वो ग्रुप में मुझे भेजा था कि ये टॉपिक्स है सिमिलरिटी को एक्सेट्रा, मैं रिपीट कर रहा हूँ वो चीज़ें, इसीलिए
ओके सर.
ऑल 45 और उस टाइम बहुत और आने चाहिए क्लास में. यू विल शेयर द लिंक in the group again and you will join again. ठीक है
जी सर.
पहले क्या किया जाता है न्यूमेरिकल टेक्सोनॉमी में? एक डाटा मैट्रिक्स तैयार किया जाता है. डाटा मैट्रिक्स कैसे तैयार किया जाता है? Only 39 people among 45 have marked their attendance. Please all to note mark your attendance here. जो डाटा मैट्रिक्स है यहाँ पे कैरेक्टर स्टेटस इस्तेमाल होते हैं. I will explain it in detail. This descriptive information that is यानी वहाँ जहाँ पे कैरेक्टर स्टेटस लिए जाते हैं, this gives a measure and we call a coefficient of similarity between every pair of taxa. इसको मैं भी समझाऊंगा अगर विंकसी इंशाल्लाह दूसरी क्लास में. taxa are clustered based on their similarity. जाने जो taxa ज़्यादा आपस में मिलते जुलते हैं वो इनको ग्रुप किया जाता है. दे आर क्लस्टर्ड टुगेदर सो दैट दोज विथ हायर सिमिलरिटी आर प्लेस्ड टुगेदर इन ए ट्री विच वी कॉल फिनोग्राम. यहाँ पर हम एक ट्री कंस्ट्रक्ट करते हैं जिसको फिनोग्राम कहते हैं, जबकि क्लैडिस्टिक्स में क्लैडोग्राम कहते हैं. इस क्लैडोग्राम में वो होता है. ये एग्जाम्पल हम नेक्स्ट क्लास में या इसी क्लास में देखते हैं. फॉर एग्जाम्पल we consider 5 species of bugs marked from A to E. बहुत सारों ने ये क्लास मेरा लिया है. यहाँ पे 5 स्पीशीज है bugs के. और फिर से सुनो, टैक्सान A, टैक्सान B, टैक्सान C, टैक्सान D एंड टैक्सान E. मैं यहाँ पर न्यूमेरिकल टैक्सोनॉमी करूँगा प्रैक्टिकली. ये 5 टैक्सा में कौन टैक्सा आपस में ज़्यादा रिलेटेड है और कौन कम रिलेटेड है. I will apply all the theory जो अभी तक मैंने पढ़ी है practically now. यहाँ पर मैं I will take character status 10 characteristics and the character status in parenthesis bracket में हम रखेंगे और noted. For example यहाँ पे आप इनकी शक्ल देखो, ये बागस है, ये बागस है, और इनकी शक्ल आप पहले सरसरी देखो, how they look and where they differ, where they have similar attributes or characters. अगर फॉर एग्जांपल यहाँ पर ये ट्रायंगुलर स्पॉट देखते हैं टैक्सान ए में, दिस इज एब्सेंट एवरीवेयर एक्सेप्ट ए. अगर ये गोल मार्कर्स देखेंगे 3, 2, 5, इट इज प्रेजेंट हियर एज वेल एंड इट इज प्रेजेंट इन टैक्सान ई एज वेल, बट टैक्सान डी में
Yes.
अलग अलग किस्म है कुछ, और टैक्सान सी में भी ये 5 मार्क्स है, बट दे आर समवट बिगर. अब इनके ये देखेंगे, फॉर एग्जाम्पल pointed nose present or absent, and in which taxa this pointed nose is present and in which absent. 4 legs आप देखेंगे, प्रेजेंट कहाँ है, एब्सेंट कहाँ है. टोज ऑन लेग्स, यानी लेग्स पे टोज, पहले में 3 3 6 3 9 3 12. और यहाँ पे टैक्सान ई में टोज ही नहीं है, टैक्सान डी में भी टोज ही नहीं है. लार्ज आइज प्रेजेंट एब्सेंट, लार्ज आइज कहाँ है और कहाँ नहीं है.
Taxon A and C.
ओ वही ना. ट्रायंगुलर शील्ड ऑन बैक प्रेजेंट और एब्सेंट, कहाँ प्रेजेंट है कहाँ एब्सेंट है. और फ्रंट लेग्स स्ट्रिप्ड, फ्रंट लेग्स में स्ट्रिपिंग है यानी मार्किंग है, और कहाँ पे है कहाँ नहीं है. ई में मार्किंग नहीं है फ्रंट लेग्स पे, लेकिन सी में भी नहीं है और शायद बी में भी नहीं है, ए में है. मैं इसको थोड़ा मैग्नीफाई करता हूँ डायग्राम को. आई विल मैग्नीफाई इट ए बिट लिटिल बिट बिगर. तो आप देखिए क्या है. और 5 स्पॉट्स ऑन द बैक, कहाँ पर प्रेजेंट है कहाँ पर एब्सेंट है. पाल्पस ऑन पोस्टीरियर एंड प्रेजेंट एब्सेंट. एंटीने थिक प्रेजेंट एब्सेंट. एंटीने कवर्ड विथ hairs so that they look stripped, present or absent. This information now, which is written in theory from point number 1 to point number 10, that is, it is written in simple English, pointed nose, present, absent. We can convert this information into a table. तयबा अटेंडेंस कर लेना और सारे लोग फिर से ज्वाइन करेंगे. अटेंडेंस हुई सबकी? जिनको जरूरी नहीं है अटेंडेंस करना, अरबिना तो गाइड पकड़.
Yes, sir.
गाइड छुपा कर तो 1 मिनट आप वेट करो, मैं आपकी अटेंडेंस. We can this information, point number 1 to point number 10, pointed nose present absent, 4 legs present absent, antennae thick present absent, we can tabulate it. हम इसको टेबल फॉर्म दे सकते हैं. दिस इज, यहाँ पे हम इस कॉलम में लेंगे टैक्सा A, B, C, D and E. नाउ इस कॉलम में लेंगे कैरेक्टर स्टेट्स 1 2 3 4 5 6 7 8 to 10 अंडर 1. यानी कैरेक्टर स्टेट फर्स्ट क्या था, कोई मुझे बताए. कैरेक्टर स्टेट
Pointed nose. Yes.
पॉइंटेड नोज. यहाँ पर देखेंगे हम कहाँ पर प्रेजेंट है कहाँ पर एब्सेंट है. ये सब में प्रेजेंट है, सो इट इज प्रेजेंट इन A, B, C, D, E. इसी तरह कैरेक्टर स्टेट सेकंड. व्हाट इज कैरेक्टर स्टेट सेकंड? इट इज
4 legs.
4 legs. It is present in
Present in uterus.
it is present in everyone. It is present in everyone.
Yes.
इसी तरह कैरेक्टर स्टेट 3 It is present in
Those on legs.
Those on legs. It is present on 3 only.
character citate 4. It is present in A and
And
C
C.
only.
Yes.
After doing this exercise, मैं आपको एक अहम जगह पर पहुंचाता हूँ. आप एक मिनट, ये ज्वाइन अभी हो रहे हैं, शायद इन्होंने क्लास छोड़ दी थी. कुछ लोग ज्वाइन हो रहे हैं. अरबीना, इनकी अटेंडेंस आप ले लेना.
Okay, sir.
मेरा मतलब था कि ये क्लास खत्म होने के बाद आप यू विल ज्वाइन अगेन यूजिंग द सेम लिंक ताकि हम आपकी सारी परेशानी दूर करें. इरम ज़रा अनम्यूट करो. इरम, आप फिर से ज्वाइन करेंगे. अभी शायद है कुछ